Now that you have macroscopically inspected the unknown bacteria by using the streak plate method and a sterile broth, it is now time to move on to the final I of the 5 I's of microbiology, which is identification. Yay! In order to identify a bacterium, you need to do biochemical testing on the bacteria first, and the first experiment with biochemical testing that we will be using is doing a phenol red broth sugar observation. For the phenol red broth sugar observation, there are a few items that you will need. You will need a bacto incinerator, inoculating loop, your unknown bacteria from your streak plate method that we had just done, your phenol red broth. Sorry it looks a little fizzy, I used juice. It's not supposed to be fizzy in a real experiment, but just to get the color, I used juice. And always make sure that you have new pair of gloves on, your goggles, and your lab coat. The first thing that you're going to do is you're going to sterilize your inoculating loop. To do this, put this in the bacto incinerator for about 5 to 10 seconds. Once you've done that, allow the loop to cool off. Once the loop has cooled off, obtain a isolated colony from your unknown bacterial culture that you had from your streak plate. Obtain one of them, close the plate back up, and then what you're going to do is you're going to take your phenol red broth, remove the cap, so, and then what you're going to do is you're going to stick your inoculating loop into the broth, like so. Take that out, put the cap back on, put the test tube back on the test tube rack, then sterilize your inoculating loop for 5 to 10 seconds. Once you've done that, put it on your bench top. Now that you've done that, take your phenol red broth and put it in the incubator. You want to incubate this culture for about 24 to 48 hours at 37 degrees Celsius. Once you put your culture in the incubator, you're going to want to disinfect yourself and your workspace. To do so, first, use germicide to decontaminate your workspace. You're going to spray it all over, but before you do that, you'd move all of these materials out of the way. Spray the germicide all over, and then wipe it down with paper towels. And then those used paper towels you would throw away in the biohazardous waste container. Once you've disinfected your workspace, you can now remove your gloves. You would also put your gloves in the biohazardous waste container, and then go wash your hands. After you wash your hands, put on a pair of new gloves because you will be conducting the next experiment.